The chapter in Genetic Twists of Fate mentions Avery, MacLeod, and McCarty, and their experiment is really a major step forward in understanding that DNA was what's called the transforming factor or the transforming principle. And they mentioned that Avery, MacLeod, and McCarty worked with strains of bacteria, specifically pneumococcus, and this is the strain of bacteria that causes pneumonia. And so there were two strains of pneumonia that they worked with, the S strain and the R strain. The S stands for smooth, and this was a descriptive term of the bacterial cell, the way it looked under the microscope. And the smooth strain was pathogenic, in other words, capable of causing disease. The rough strain had a different coat appearance on it or surface appearance to it, and the rough strain was not pathogenic. In other words, if you take mice and inject them with the R strain, the mice will live. But if you take mice and inject them with the S strain of bacteria, they will die of pneumonia. And so the work of Avery, MacLeod, and McCarty was really to try to determine what was this factor that was initially discovered by a scientist named Griffith that was this transforming factor or transforming principle. So what they did was they took the S strain of bacteria and they exposed it to different types of enzymes. Now, this is a very simplistic overview of it, but it gets the point across. It's very creative. If something ends in an ASE ending, it's an enzyme. And one of the things enzymes can do is speed up reactions that break things down. So, for example, DNase is an enzyme that breaks apart DNA. RNase breaks apart RNA. Lipase breaks apart lipids. Protease breaks apart proteins. Amylase breaks apart carbohydrates. And so the idea was that this transforming factor had to be a biological molecule. And so if we take enzymes for the common biological molecules that were known and digest fractions of the S strain of bacteria, then exposed it to the R strain and injected it into the mice, then whatever enzyme prevented this transformation of the R strain into the smooth strain, that would be the transforming factor or the transforming principle. Because remember, what Griffith had done was taken heat killed smooth strain, mixed it with R strain, injected into mice, and it killed the mice. So that meant something was transforming or changing the R strain. So they set up the experiment using DNase, RNase, lipase, protease, amylases, and other things. They exposed the digested S mixture with the R strain, and then they injected it into mice. And the only situation in which the mice lived was when DNase, this mixture of S strain bacteria, had been exposed to the DNase. And that's what provided the evidence that it was DNA that was the molecule of heredity or the transforming factor that converted the R strain, which normally didn't cause pneumonia, into the S strain, which did cause it. So I hope this little extra figure adds some clarification to what was mentioned in the chapter.